had the opportunity to connect with Dr. Shibani Pati, who is a professor in the School of Medicine at UCSF. Shibani received her MD, PhD from the University of Maryland School of Medicine and has a deep interest in vascular biology. In this interview, we were able to gain some insights from her work using MSCs as a cellular therapy to treat ARDS and listen to her perspectives on the challenges and future for MSC therapy. Hi Shibani, it's such an honor to be speaking with you today. Thank you for taking the time to be with us. I really wanted to take this opportunity to hear some updates from you since COVID. So I know that you are a vascular biologist and your research is really focused on vascular endothelial permeability, and the abnormalities of these are often observed in conditions like traumatic brain injury or even ARDS. Your lab has been very active in looking at some MSC-based cell therapy as a treatment modality to treat these patients. So I was just wondering, how has COVID sort of affected your work? And maybe you could share some updates with us here.
So thank you so much. I'm so happy to be part of this program that you've organized. And as I told you before, I've known RoosterBio since its inception and have sort of followed your progress along the way in the field of MSCs. So I'm really happy to talk about them. But it is a great question. I mean, one of the aspects is that really, and if you talk to colleagues, they'll say that there's positive aspects and negative aspects. And for me, I'll start with the positive. It's hard to find positive in a pandemic, but one of the positive aspects of the shutdown and the limited capability to work in lab for me, and in particular, actually has been my ability to really focus on things that I would normally not be able to complete, or I was just too busy to complete. So, we spent a lot of time trying to finish papers, submitted papers, we've written a number of grants in this time. Some of them have been COVID related to MSCs, things that would require focus and attention, and being at home kind of has helped out. On the negative side, my institution, until just last week, was at 25% of our workforce. We were shut down for a couple of months, and we went back in early May. And 25% of our workforce in the lab is really, as you know, for lab work, is very limited.
Yep.
And so we kind of kept things going as kind of the sort of a back burner, level. But now that we've been increased to 50%, I think things will change. And we had, it was tough on the guys in the lab. Some of them have limited amounts of time. Some of them are residents, some of them are postdocs. And so they don't really have the luxury of just waiting things out. So they were working in shifts. And for example, one of our scientists, she has little kids. She was coming in at 4:00 in the morning and leaving, in the early, later morning hours before noon. And so, these are the kind of things that were difficult. And what's difficult actually is we've had some new folks in the lab, and I'm sure everyone shares this, is that training them is difficult because we have to maintain a social distance and
Right.
in a safe, and teach them in a safe way. And as everyone knows, when you're working in lab, you really sometimes want to be leaning over the shoulder of the other person, like when you're in the tissue culture room or when you're on the bench, and because you can't really see, so unless you're close to the person. So that's been difficult. And on a personal side, I mean, I think just speaking as like a mom and having my family issues to deal with, challenges for me personally have been dealing with work, and also the issues that arise at home with kids being on school online, doing internships. I also have my elderly parents at home, and so we're always extra worried and careful about them. So I think it's kind of it's an interesting time because we're mixing both our home issues with our work issues. I think women probably, I will be as bold to say this, probably have it harder than a lot of others who are struggling through this in terms of trying to juggle everything from work to home life. So, but we try to stay positive. And the one thing I tell my lab consistently is like, any progress we make in the lab, say we even a small experiment and it actually gets done and it's pulled off well, it is progress, and it is a reason to be happy and to celebrate that it actually got completed. So so yeah, so those are kind of the plus minuses. I mean, I think we've learned a lot, and we've learned also how to work together in a good way.
Yeah, thank you for sharing that. I think that is so true, with everyone that I've spoken to in different aspects of their work as well. Like the work-life balance is so challenging, and just, you know, keeping the positive energy and just positive reinforcements to help each other get along, right, and get past these challenges together. I think it's really the right way and right approach of doing things. So thanks for sharing that, Shibani. I think that's really insightful. So on the research side, I also wanted to ask a little, I guess, dig in a little bit and ask, do you think COVID has presented more or less opportunities for your research as far as like maybe funding or other research collaborations? Would they maybe postpone perhaps because of the COVID situation?
Oh, so those are great questions. Well, it's interesting, so my background, my PhD was in virology at the University of Maryland, working with Bob Gallo's group on HHV8. So I've always sort of been interested in viruses and the pathogenesis of viruses and diseases they cause. So when SARS-CoV-2 came out, and sort of the biology started to emerge, we were following it. And I think what struck me, which sort of drew our lab's interest into it, was that there is, as you mentioned, and I didn't get into what our lab focuses on, but we are primarily focused in the area of vascular biology, endothelial dysfunction, clotting dysfunction, and trauma-related applications. And we're also very interested in COVID-19 related pathology because, as you know, it piqued our interest. If you really sort of get a feel for the presentation and the multiple papers and descriptions, and even speaking to colleagues of mine who literally are, their offices are right next to mine taking care of COVID-19 patients, is just sort of the systemic nature of the disease. The aspects of multi-organ failure, the thrombotic components of severe disease, all sort of sing to us as there might be an underlying systemic endothelial dysfunction, much like what we see in trauma, and in many ways can be a therapeutic target to address for treatment. And so when this started to emerge, we formed some partnerships and wrote some grants, and also thought papers that it would be interesting to test out some of the therapies that we know target the vascular endothelium and organ failure. And one of them, of course, just to bring it up, is MSC. So that has presented some interesting opportunities, and we've partnered up with some of my these are like blasts from the past colleagues from earlier, who are virologists who are actually making viral constructs of SARS-CoV-2. Some of them are making constructs of the different proteins and sharing them with us. So one of our goals is to be able to sort of understand if any of these particular proteins contribute to endothelial dysfunction and injury. And of course, MSCs are in there. Like I said, we've written for support to see if we could test out MSCs. And we're also part of a grant right now, which is testing the role that MSCs could be used as therapeutic in ARDS. And it turns out this is the prime organization is actually UCSF. It's being run by Dr. Michael Mathey, but it's a multi-center placebo-controlled randomized trial, which was initially, as I mentioned, meant to enroll standard ARDS patients who have ARDS secondary to trauma or to sepsis. But because of COVID-19, the majority of the patients they are enrolling are actually COVID-19 patients. So they are getting MSCs or placebo. So we don't know, and of course we'll have to wait till the end of the trial before we find out. So I think it's presented some new opportunities, but also hindered some of the possible collaborations that we had started to embark upon before the pandemic hit. So I think, it just like anything, there's a positive and a negative side, but we're trying our best to be positive.
Yeah, absolutely, absolutely. Yeah, awesome. So in terms of, there's been a lot of discussions, a couple papers that even talk about the success of these cellular therapies really hinge upon various factors, right? So I wanted to hear your perspective as far as where do you see the biggest challenges in terms of ensuring success of these MSC-based therapies for COVID trials, and what do you think we need to do to overcome these challenges?
Oh, so that's a very good question. So the first question that rises to mind is that, um, it's because this is so new. We are only hypothesizing based on what we know from our preclinical data and clinical data in other applications that MSCs could potentially be a therapy for COVID-19. Now, there is some data coming out, and there are definitely multiple trials worldwide being run. But there was early on some very small trials from trials that were run in China that demonstrated that MSCs might actually have some therapeutic potential and benefit in COVID-19 patients. So, and then from everything we know about their mechanisms of action, we would hypothesize they would definitely have an effect in patients infected with SARS-CoV-2 or that are COVID-19 patients. But the one thing that I'll tell you that sort of came up and I was just talking to you about this earlier, a challenge is that so if you think about it, it's hard to scale up MSCs for the masses, and so dose production is definitely an issue. So, of course, there's all these other issues that come up, timing, therapeutic window, what target patient population to give it to, when, what would be the most optimal time? Do we need one dose, two doses? Those things, aside from that,
Right.
If you think about it, to be able to have enough doses to really treat the numbers of patients that are coming in, depending on what your target patient population is who have COVID-19, that I think is something that we've been discussing as a group. And, BARDA, have you heard of BARDA, which is a organization that's very interested in trying to support these sort of national disasters? They had announced that they were going to get into cell therapies for COVID-19 induced ARDS, but they actually changed their mind. Now, I don't know exactly why, but I think in part there might have been some aspect of the inability to scale up and bank adequate numbers of doses. And I think that that's probably one of the major problems. And that's something that exists even now for even the trials that we're part of. We barely have enough doses. We're always waiting for the next set of doses before we can even embark. Yeah. So I think that that's really important. There's going to have to be a really dramatic change in cell production and high quality cell production. That's another thing I wanted to bring up. Actually, I'm happy you raised this question. Is that so? If you look on clinicaltrials.gov, you see there are so many trials testing MSCs in various applications, and most of them have not shown. Probably all of them, pretty much, have barely shown any efficacy at all. And we have started to believe that a lot of it has to do with the quality of the cells. I think the quality and really understanding quality and potency. And so I can get in a whole. Basically, I have an hour-long presentation on cell potency for MSCs. But as you know, that's critical too, right?
Yeah.
And it's going to differ by what application you're putting it into. There might be a particular subset of MSCs that are ideal or donor MSCs that are ideal for a particular application and not for another, just depending on what the potency markers are and the mechanisms of action are. So that's kind of, I think those are the struggles, I think. And
Yeah.
I don't think they're insurmountable. I
Yeah.
think, one of the areas RoosterBio has gotten into, I know, is 3D bioreactors and production using these, these sort of large setups. That's kind of, I think, where this has to go. There has to
Yeah.
be adequate doses. Yeah.
Yeah, I think you would
So,
definitely hit it on the nail right there, Shibani, because that has been something that we've been very much focused on is the scalability, but also maintaining quality is just absolutely critical. It's not just the output, but the input materials. What you start with, right, has to be high quality products as input materials in order for you to get high quality output materials as well. So thank you for sharing that. I think we're definitely in sync and aligned with you on that one as well. Yeah.
yeah.
So I always love to hear about your past, and I would definitely like to hear how you got into this area of research. What got you excited, and why did you decide to get a MD PhD? I guess that's a lot of work.
Oh, well, yeah. I mean, actually, it's interesting. So I don't practice clinical medicine, and I always knew I wanted to be a scientist. But I think that I went to college, and I was actually a physics major, and it actually goes back to then. And I just had the most wonderful undergraduate lecturer. His name was Arnold Levine, and he was one of the discoverers of P53, and he had an introduction to molecular biology course, and like I took his course and I was like, this is my calling. And so then I was fortunate to have really great mentors. And um, when I went to medical school, I had every intention of focusing on research. It was really what I wanted, a medical background to be able to apply it
Mm.
to pertinent areas, things like translational medicine. That's the term that emerged after I went to medical school.
Yep.
So, but yeah, I mean, an MD PhD, I think it just depends on the field. There's so many MDs who do amazing work. You don't have to have a PhD to
Sure.
be able to embark upon really fundamental science and medical work. Biomedicine, but it was good for me. It was a great experience for me, and it was a great exposure. And then the other aspect of it that really has been just almost like a real blessing for me is that I have outstanding partnerships and collaborations and friendships with clinical uh, uh, um, surgeon scientists and
Mm.
physician scientists who are in the area of actually treating patients and translating from bench to bedside. So that's really what our aim is. And I'm sort of a bridge between the two worlds for in some of these partnerships. And so I think I feel that, I've been very lucky to meet the right people, to get the right mentors. And I think one thing I would say to anybody is I think no matter what field you're in and how old you are, you still need mentors. I go back to my old mentors all the time to ask them questions when I'm stuck on something or don't know what to do for career choice or things like that. You just need really good mentorship and you have to seek it out. It's not sometimes you're lucky it'll fall on you, but if you seek it out, those people will become your go-to people who you spend a lot of time talking to and asking guidance from. So, yeah. So, I mean, I think everyone has their own road of how they got to where they are. But for me, I think I just always wanted to do research. In science. So, and it just, it's kind of like just meeting the right people at the right time. I met John Holcomb in Texas at the University of Houston, and he sat next to me at it's just serendipitous at a lunch, and he goes, I'm really interested in endothelial cells, and I'm a trauma surgeon, and I'm just like, well, we should talk, and then it just started from there. Yeah, I mean, it's really interesting. It's fascinating.
Yeah.
So,
Yeah, I think that's very true. I think sometimes it's also serendipity, who you meet, and the paths that you cross with each other. And and just grabbing those opportunities, right? So I think that's really wonderful that the path has led you here and led you to RoosterBio as well. That we became friends with you for such a long time. So do you have any advice? I'm sure there's a lot of young aspiring scientists or clinician scientists out there who may want to follow your path. What advice would you give to these young people out there who are, looking to develop their careers?
Oh, I guess I would tell them to follow their passion, to 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 work in something that they love to do, to pick the right thing. And it shouldn't be something that you think, it shouldn't be based upon, how much money you're going to make or any of that. It should be that you think that this job is going to be something that you love doing.
Mm,
So I think that's always been the guiding principle for me. And then of course the other stuff is important, location and salary and all that stuff. But the first thing is make sure you love it. Like it has to be something that calls to you, that you wake up to, not every day maybe, but you're like, I love my job. I
right.
love what I'm doing. Yeah.
Right.
So that's kind of
Right.
it. And that's basically, yeah, I'd say. And then find a good mentor. I can't stress the
Yeah.
importance of mentorship. Find a good mentor and stick to that person. Yeah, you could have a couple of mentors too, just
Yeah.
not
Yeah.
too many.
That's great. That's awesome. Yeah, I think
Yeah,
that's really good advice. I think, you have to be actively looking for the mentor and also seeking out to them, right? I think that's definitely very true because you can't just expect people to come to you. You really got to, go out there and seek out. So that's, yeah, I agree with you on that one. So you've done a lot, Shibani, and accomplished a lot in your career. What would you say you're most proud of? What makes you happy, I guess, looking back?
oh, I feel like I have so much more that I want to accomplish, but I think what I'm most proud of is the partnerships that I've formed with the clinic bridging. It really, I was brought this up before, is really the translational aspect of the
Mm,
work that we do. Is that we do have our roots in basic science and fundamental biology, but most of what we do is meant to be translated
mm,
soon and rapidly from bench to bedside. And so we try to engage in product and projects that and partnerships with people that become friendships actually. And then you form these groups where you kind of brainstorm together. And I think that that is something that I would say I'm most proud of. And I don't know if I'd use the word proud, but I'm very happy I've been able to do this. And CTAC, which is a meeting that you've been to, that we started early in 2012 for cell therapies and trauma and critical care, has been actually a partnership was formed between RoosterBio, the DoD, and BioBridge Global, I think,
oh,
at one of the CTAC meetings too. So I feel like we've brought people together, and
yeah.
and I think without good teamwork, we won't go anywhere. I mean, it's all facets that are needed to be able to launch anything from
Yeah,
bench to bedside. So
yeah,
yeah, that's kind
yeah,
of what I would say.
yeah.
Yeah.
No, this is. I think this the CTAC meeting that you organized. I was very impressed with it. It was the first time I attended last year, but I really like how focused the topics were, how close knit, the environment and the people. So you could really feel that community and the ecosystem. So I was very impressed. I was very impressed.
Oh yeah, no, it's great that you came. Brio's been there from the start. I think
Yeah,
John's spoken at it like every single
that's
time.
awesome. That's awesome.
So yeah. Well, thank you.
Yeah. So, well, I think maybe last question for you. Looking ahead, what are you most excited about, and what are the biggest bottlenecks maybe that you think we should be overcome? We should overcome or look at.
When you say excited about, are you talking about for my own research or just in general?
Just in general, like regenerative medicine and cell therapy, I guess. Yeah. Sorry.
Oh, we need to see efficacy. We want some of these phase 3 trials to get to show some positive signals that we can actually get something into clinic and it become a routine part of patient care. It makes a difference,
Yeah. Yeah.
want to make a difference.
Yeah.
Yeah, that is what, and I think we're right on the horizon of it. I think we shouldn't give up. And I know MSCs have gone up and down in popularity, but I think that they have a lot of potential. So they're going to they're still in the running.
Yes, I agree with you. I think I'm very optimistic as well. So we'll definitely get to impact in the next 5 years. I'm praying for that too, for sure. Awesome. Great.
Absolutely.
Well, thank you so much, Shibani, for your time today. Any last words for the audience that you'd like to share before we kind of close this session?
No, I'm just very happy to have been part of this. I hope that in better times that things will move faster and that we'll be able to, like I said, see a difference that cell therapies can make in patients. So thank you again for inviting me.
You're welcome. Thank you so much for your time. Really appreciate you sharing all your insights and everything, your experience with us. So thank you.